Hi everyone, my name is Dr. Carratt, and I'm going to talk today about the coronavirus. Specifically, we're going to talk about the biology of SARS CoV-2, which is the virus that causes the COVID-19 disease. I'll begin with a brief introduction. Again, my name is Dr. Carratt, and I'm excited to record this presentation for you because I loved the biology classes that I took with Dr. Wallenstein at AMHS. I have a Bachelor's of Science in Biology and a PhD in pharmacology and toxicology. I spent 4 years researching respiratory diseases at the University of California, Davis, and I'm currently studying leukemia at Oregon Health and Science University in Portland, Oregon. If you have any questions about a career in science or about the content presented in this lecture, feel free to email me at carratt@ohsu.edu, listed at the bottom of the screen, and I'll show my email again at the very last slide. So let's begin, and we're going to learn about the biology of SARS CoV-2. Let's start off by clarifying some jargon. SARS CoV-2 is the virus that causes COVID-19, which is the respiratory disease that is spreading throughout the world. The name COVID-19 was derived from the year that the disease was first detected, 2019, and using the letters from coronavirus disease, COVID. Let's take a look at the name of the virus in the same way. SARS, severe acute respiratory syndrome. This virus causes a respiratory disease with pneumonia-like symptoms. CoV. CoV stands for coronavirus, or the scientific family name Coronavidae. The word virus comes from a Latin word meaning venom and describes a tiny agent that causes infectious disease. Coronavirus is a family of viruses that got its name from its appearance, which is similar to a crown. The number 2 at the end of SARS-CoV simply indicates that this is the second virus of its kind to be seen in the world. The original SARS-CoV was the virus that caused SARS, severe acute respiratory syndrome. While SARS-CoV also starts with the term SARS, what we're describing from SARS-CoV-2, we're terming a coronavirus, or simply COVID-19. This can be a little bit confusing in the media, since both viruses are very similar in structure, but they cause two very different diseases. Both of these SARS coronaviruses have spread throughout the world in different epidemics. COVID-19 is now a pandemic and has spread further than SARS-CoV-1 did. The original SARS epidemic in 2002 spread to 37 countries and ultimately impacted about 8,000 people. This is a map of how far the epidemic spread back in 2002. Again, this disease was caused by SARS-CoV or SARS-CoV-1. This beta coronavirus caused severe acute respiratory syndrome or SARS. The new SARS-CoV, SARS-CoV-2, is a novel coronavirus that causes coronavirus disease 19, or COVID-19. Here we see a diagram of SARS-CoV-2 with each of the proteins that are critical for its function labeled. Generally, coronaviruses are positive-stranded RNA viruses. Under a high-powered electron microscope, coronaviruses have a crown-like appearance due to the presence of spike glycoproteins on the surface of the envelope. Here we can see those labeled as spike glycoprotein S. I guess I can see it. Members of this large family of viruses can cause respiratory, hepatic, and neurological diseases in different animal species. This includes camels, cattle, cats, and bats. To date, there have been 7 human coronaviruses capable of infecting humans. Some of these human coronaviruses were identified in the mid-1960s, while other viruses like SARS-CoV-1, which we talked about, which causes the SARS epidemic, were identified much more recently. SARS-CoV-1 was 2002, and obviously COVID-19 was in 2019. Coronaviruses are generally responsible for about 5 to 10% of acute respiratory infections, and are generally no more severe than your average cold. Each SARS-CoV-2 viron is approximately 50 to 200 nanometers in diameter. This is similar to rabies, which is 75 by 180 nanometers, influenza A, which is approximately 100 nanometers in diameter, and HIV, which is 120 nanometers in diameter. The structure of the SARS-CoV-2 virus has multiple parts. We'll focus on just 3. Inside the virus is single-stranded RNA, which is the genetic material of the virus. The viral envelope encapsulates this genetic material. On the surface of the viral envelope are a number of proteins, but the most important by far is the spike glycoprotein, labeled spike glycoprotein or simply S in this figure. The 2019 coronavirus has a mutation in this S protein, the spike glycoprotein. These S proteins, these spike proteins on the coronavirus, bind to an ACE2 receptor, an angiotensin converting enzyme 2 receptor. This receptor is a transmembrane enzyme. That means it goes through the cell membrane on specific types of cells. It serves as a main entry point to these cells for coronaviruses and is also the entry point for SARS-CoV or SARS-CoV-1. The binding of the spike 1 protein of SARS-CoV and SARS-CoV-2 causes the enzyme, the ACE2, to endocytose into the cell, resulting in the translocation, the movement of both the coronavirus and receptor to the inside of the cell. Now, these ACE2 receptors are located in actually a variety of places in the human body, including the lungs, arteries, heart, kidneys, and intestines. But what we see here is that they're also, most importantly, located at the respiratory gas exchange surfaces. This is important because SARS-CoV-2 causes a respiratory disease, and therefore it's really important to understand why this coronavirus coming into the lungs matters. When the SARS-CoV-2 spike protein binds to this receptor on cells in the alveoli, it causes a series of downstream inflammatory problems in the lungs. In this animation, we see that the S spike of the coronavirus allows the virus to bind to the cell surface receptor ACE2. In the lungs, cells with these receptors are located primarily at the gas exchange surfaces. When the coronavirus binds the ACE2 receptor, it is endocytosed. Once inside the cell, the virus releases its genetic material, single-stranded RNA, into the cytoplasm. With the viral RNA inside the host cell replicates the viral genome using RNA-dependent RNA polymerase. Concurrently, the host cell begins synthesizing all the structural proteins needed to form a new virus, from the proteins that make up the envelope to the all-important S proteins. These newly synthesized proteins combine and incorporate the replicated single-stranded RNA to form a new viron. The stress of viral reproduction on the ER leads to apoptosis or programmed cell death. So let's talk a little bit more about what this replication and cell death means in the lung. Here's an image that I showed before where the coronavirus is binding to a cell in the alveoli at the gas exchange surface. I'll focus on these cells for the remainder of the presentation because the pathology, the laboratory analysis of patients with COVID-19, suggests that these cells are important in causing most of the disease symptoms. However, I do want to mention that the lung is made up of many, many types of cells that each have unique functions. There are a number of cells that SARS-CoV and SARS-CoV-2 can bind to and infect, and the literature suggests that SARS-CoV-2 can infect other lung epithelial cells, which are shown in brownish gray here, and usually affect the more mature ciliated cells. Okay, back to the alveoli. While there are many cells in the lungs that can have ACE2 receptors, 83% of the ACE2 expressing cells are type 2 pneumocytes, or alveolar epithelial type 2 cells. Type 2 pneumocytes are located in the alveoli where they secrete surfactant. Surfactant is very important for normal lung function and gas exchange. As mentioned earlier, the stress the virus puts on an infected cell usually results in cell death. In the alveoli, the loss of type 2 pneumocytes, these surfactant-secreting cells, has consequences. The main purpose of surfactant is to lower the surface tension at the air-blood interface within the alveoli. This keeps these small alveolar air sacs from collapsing when an individual exhales. Loss of surfactant can lead to alveolar collapse, and at a minimum, it increases the work of breathing. You have to work extra hard to inhale because against this increased pressure, that process becomes more difficult. This results in shortness of breath in a patient. This is an image showing a healthy alveolus on the left and an alveolus with acute lung injury on the right. Acute lung injury, like what we see when the coronavirus kills lung epithelial cells, impacts inflammatory signaling. Lung inflammation is the main cause of life-threatening respiratory disorders at a severe stage. Inflammation is triggered when the damaged and dying type 2 cells activate pro-inflammatory macrophages and granulocytes. Macrophages are a type of white blood cell that are part of your body's defense mechanism and are part of the cause of airway inflammation in asthma. They function by 1. Engulfing foreign substances or cells, or 2. By secreting pro-inflammatory signals or mediators. Some of these mediators can cause vasodilation, and the capillaries at the interface of blood and air become more permeable. This causes fluid or plasma to leak back into the interstitial spaces, the lining of the lung, and even into the alveoli itself. It should be noted that the inflammatory responses can cause a lot of oxidative stress in the lung. The accumulation of cellular debris in the lung essentially paints the walls of the lung, making it more difficult for gas exchange to occur. This ultimately leads to pneumonia-like symptoms in COVID-19 patients, though the severity of symptoms can be variable. In mild and moderate cases, the patient's lungs and airways swell and become inflamed. You might be able to see some abnormalities show up if you perform a chest X-ray or CT scan. About 14% of COVID-19 cases are severe, with an infection that affects both lungs. As the swelling gets worse, the patient's lung fills with fluid and debris. The air sacs fill with mucus, fluid, and other cells to fight the infection. This can make it harder to take in oxygen. In critical COVID-19, which is about 5% of total cases, the infection can damage the walls and linings of the air sacs of the lungs. As your body tries to fight it, your lungs become more inflamed and fill with fluid. This can make it harder for them to swap oxygen and carbon dioxide. In the most critical cases, your lungs need help from a machine called a ventilator to do their job. Why some people get sick and others are asymptomatic or not presenting with classical symptoms is still a little unclear in the literature, but it likely has to do with the degree to which inflammatory signaling is triggered. We know that the progression from early stage COVID-19 to late stage disease is marked by a dramatic increase in inflammatory mediators and an influx of immune cells to the alveoli. To summarize, SARS CoV-2 is the coronavirus that causes COVID-19. Its most notable feature is the S protein or the spike protein. The spike protein is responsible for allowing the virus to attach to and fuse with the membrane of the host cell. The respiratory symptoms of COVID-19 arise as a result of the virus binding to the receptors that are located at the gas exchange surfaces of the lungs. The alveolar cells that die from the stress of reproducing the virus causes a loss of surfactant, making it more difficult for patients to breathe. The virus also triggers a series of inflammatory responses in the lung by activating macrophages. These inflammatory responses affect the vascular system, causing an influx of fluid to the lungs. Now that you are all experts on how SARS CoV-2 enters a cell and causes symptoms, see if you can understand these two strategies that have been proposed for the treatment of COVID-19. The first strategy is that you can target ACE2 receptors. This will block the ability of coronavirus to bind to the receptor by giving the receptor something else to bind to. If the receptor is occupied, it can't accept binding from the coronavirus. The second strategy is that you can target the receptor binding domain of the coronavirus itself. It's been proposed that you can make antibodies against SARS-CoV-2 spike proteins. If you have antibodies bound to the spike proteins are not able to bind to the ACE2 receptor. That's it. Thanks for listening to my presentation. Again, my name is Dr. Carratt and my email is listed here. Feel free to reach out to me if you have any questions about the content that I covered. I'm also happy to talk to anybody about careers in science or what it's like to work at a research hospital. Thank you again.